Hey there, and welcome back to Ethical Controversies in the Medical Field. Today we talk about therapeutic cloning. I'm your host, Colin Peterson, and today we're diving into one of the key techniques behind therapeutic cloning, something called somatic cell nuclear transfer, or SCNT for short. It sounds complicated, but don't worry. I'll break it down step by step. So, somatic cell nuclear transfer is a laboratory technique where scientists take the nucleus of one cell and transfer it into another. But it's not just any cell. A somatic cell is taken from a person. It can either be a skin cell, liver cell, stomach cell, anything that has a person's complete DNA in the cell. Then, scientists take an egg cell, usually from a donor, and carefully remove its nucleus, which is part of that cell's DNA. Now, this is where the magic happens. They insert the nucleus from the somatic cell into the empty egg cell. So now the egg contains all the DNA from the person who donated the somatic cell. Pretty futuristic stuff, right? Now, once the new egg has the somatic cell's DNA, scientists give it a little push, usually a small electric shock. This kickstarts the cell into dividing, just like a fertilized egg would. If all goes well, the egg begins to grow into a blastocyst, which is an early stage embryo. This blastocyst has stem cells that are genetically identical to the person who gave the somatic cell or skin cell. Now, why is this such a big deal? This is such a big deal because these stem cells that come from the early stage embryo can be used to grow tissues like nerve cells, heart cells, or even insulin-producing cells that match the patient's DNA. This could be life-changing for treating diseases like Parkinson's, diabetes, or spinal cord injuries without having to worry about rejection. Immune rejection is a big problem. When you get a transplant, even if it's a near match, your immune system might see those new cells as foreign invaders and try to destroy them. To lower the chances of rejection, patients usually have to take immunosuppressive drugs for the rest of their lives. These drugs weaken the immune system so it won't attack their new tissue, but it also means they're more likely to get infections and other illnesses. And even with medication, sometimes the body still rejects the transplant. That's where somatic cell nuclear transfer and therapeutic cloning can be game changers. Because we can grow custom matched tissues using a person's own DNA, we might be able to skip the rejection problem altogether. Another problem that therapeutic cloning can solve is patients on the waiting list for new tissues. Imagine you're a patient with a failing organ. Maybe your kidneys aren't working anymore, or you've had a heart attack that left your heart damaged. In many cases like this, you might be told that you need a new tissue transplant or even an organ transplant to survive. You are put on a waiting list, but here's the hard truth. There aren't enough donors to meet the demand, so some people wait months or even years for a match, and sadly, not everyone makes it through that wait. Okay, let's talk about something a little heavier, the ethical side of therapeutic cloning. We've talked about how somatic cell nuclear transfer can help create genetically matched tissues and how it could help patients avoid rejection. It sounds amazing, right? But there are some people who can be against this. When scientists use SCNT, they create something called a blastocyst, which is a very early stage embryo. Although it's not implanted into a womb or meant to grow into a baby, some believe that creating and destroying embryos, even for medical research, is ethically wrong. It raises questions like, is it okay to create human life just to harvest cells? And where do we draw the line between research and human rights? As well as, should embryos have moral or legal status? These are tough questions because people of different cultures and religions have different views on when human life begins. Some believe that life starts at conception, the moment the egg is fertilized, or in this case, when the nucleus is inserted into the cell and it starts dividing. Others believe that early stage embryos don't yet count as life in the full sense, especially if they were never intended to develop into a person. Now, there are very valid concerns about this, such as if we can clone tissues, what's to stop someone from trying to clone a whole person? Therapeutic cloning is very different from reproductive cloning. The goal of therapeutic cloning isn't to make a baby clone, it's to grow cells that can help people heal. But that line can feel blurry for some, and it makes the public debate even more intense. Okay, now let's talk about the difficulties that come with therapeutic cloning. Remember how I said it takes a human egg to create a cloned embryo? Well, the process of harvesting a human egg is very difficult. It has a high mortality rate for people who want to donate these eggs. Additionally, the animals produced by cloning in lab tests have seemed to have major difficulties and struggle to live. In the most famous cloned animal, Dolly the sheep, this animal had to be euthanized at 6 years age because she was frequently suffering and battled many diseases throughout her life. For today's podcast, we have a very special guest. Dr. Kondik is the author of an amazingly well-written article, When Does Human Life Begin? She has researched in the development and regeneration of the nervous system. In 2002, she was named a McKnight Neuroscience of Brain Disorders Investigator in recognition of her research in the field of adult spinal cord regeneration. Dr. Kondik has also been awarded the Basil O'Connor Young Investigator Award for her studies in peripheral nervous system development. Hello, and we have a special guest here with us today, Dr. Maureen Kondik, and we're going to be talking about somatic cell nuclear transfer and what the embryo is, and the difference between a human embryo and an embryo produced artificially through somatic cell nuclear transfer. How are you doing today?
I'm doing well, and I'm very happy to that you invited me to speak with your audience.
Yeah, I'm very glad I have you here today too. So a human embryo, when does a human embryo have the qualities of a human life?
It's a good question, and it's one that people have debated in a wide variety of ways for many centuries. So we've all had curiosity about our origins, about when do we start. I think many of the answers and why the topic is so confusing is that many of the answers really were based on very little factual evidence. But I'd say within the last 50 years or so, we've been able to understand and observe the beginnings of life at sperm-egg fusion. And I think there's a very strong consensus in the data that human life begins at the instant when a sperm and an egg fuse to each other.
Mm-hmm. And that's a great point. I mean, the cellular changes that undergo instantly when sperm and human egg, come together to create an embryo or zygote are incredibly different, and the processes after a zygote is formed and the composition that it creates is very obviously different from any other human cell. Now, when a zygote is created in somatic cell nuclear transfer, they have a composition distinct from a zygote produced naturally. Does this difference affect the autonomy of the embryo produced through somatic cell nuclear transfer?
Well, somatic cell nuclear transfer, or I think most people would be more familiar with the term cloning, is a very inefficient process. So typically only about 1 to 10% of all those procedures of moving the genetic material from one human cell into an egg cell, only about 1 to 10% of those experiments or those procedures will actually result in a zygote that can move forward in development. So there's obviously a lot of difference between the great majority of things you produce by cloning and a natural human embryo. But even the ones that succeed, even the ones that can move forward, we know mostly about this from animal experiments. So in that 1 to 10% where the cloned embryo will go forward in development and produce a live-born animal, those animals are very abnormal. They do not have the same characteristics that an individual produced by natural fertilization would have.
Yeah, I agree with you there. I mean, it's very difficult, and it's difficult to obtain the eggs for this procedure as well.
Yes.
And it can be a life-threatening procedure as well. And the outcome of development in a cloned zygote can be very difficult for the life of that cloned individual as it develops into a fully formed functioning organism. Now, because of those difficulties in development, would it be considered wrong to halt the embryo development at a certain embryonic stage to benefit the lives of others, let's say people who need organ transplants or tissue transplants?
Right, so many people have debated this exact question. Would it be ethically permissible to produce a cloned human zygote from the cells of a patient who needed perhaps a stem cell treatment, or perhaps needed an organ or components of an organ to address their medical condition? So then use that embryo, allow it to mature to whatever stage you need to obtain the material you need that's now genetically matched to the patient, and then destroy the embryo to obtain its cells or to obtain its organs. I think it's an attractive idea. It's one that has proven very difficult to actually put into practice in terms of producing viable cells or organs from a cloned individual. But when you're talking about a cloned human, the real question is it appropriate to make an individual in a laboratory who you know will have medical problems, the problems associated with cloning, to benefit another individual, and to use them as an instrument for the medical treatment of a sick person? I think for me, as a Catholic and as a scientist, I have great problems with that idea, turning humans into tools for the benefit of another human.
Kant, a philosopher, argues that it is wrong to use human life as a means to the well-being of another or many others. Do you think cloned embryo fits into the criteria of a human life?
I would say, from a perspective of a biologist, absolutely.
Okay.
It's all of us were zygotes at the beginning of our existence, and we went through all of the stages that a developing embryo goes through. They're very small. They're initially simple, but they're in a process of, out of internal resources, out of their own materials, out of their own programming, of building more mature stages of development. So similar to a newborn baby who is nowhere near as capable of taking care of itself or doing the things that an adult would do.
Yeah.
But that baby will, out of its own internal processes, mature into an adult. And it's the same thing with an embryo. So yes, I would say an embryo is a human person at the youngest stages of development, and instrumentalizing them for the benefit of someone else would be wrong.
Okay. So you would say that the dignity of an embryo created naturally and an embryo created through cloning, they're the same dignity?
I would say that the challenge of cloning, of course, is that you're producing an embryo that is likely to have significant deformities, difficulties, dysregulations of genes that will impact the health and survival of that person. But its dignity, simply because it has a medical condition or a disability, is the same. I mean, I would not see a person in a wheelchair as being less dignified than a person who could walk.
Yeah, I agree with you. In the case that, let's say, in the future, embryonic cloning has developed to ignore the deformities, and it grows into a cloned human person, people that argue against reproductive cloning would say that because the individual resulting from cloning has the exact genome as a parent, the autonomy of this individual will be undermined.
I find less weight in that argument, both biologically and philosophically. So we already have a natural form of cloning, which is the formation of identical twins. So when you have identical twins, they are exactly the same genetic information, and lots of studies have been done to try to address the bonds or lack of bonds between twins, and I think twins are fully autonomous. They see themselves as individuals. In the case of cloning, there would also be a great age disparity between the two twins, the cloned individual and the adult from whom the genetic information was taken.
Yeah.
And I think that to some extent it would depend on what the purpose of producing that clone was.
Okay.
If you are producing it in order to provide you with donatable organs, obviously that's going to impact
Mm-hmm.
autonomy and dignity of the cloned individual. If you're cloning it to have a child who's exactly like you, that puts a huge burden on that person. Yeah. But I don't know that it would change their autonomy. They would be active agents, capable of free will and capable of making their own choices.
That's a great point right there. Do you think that in the case of zygote development and the use of somatic cell nuclear transfer, there might be a social bias or social bias against it, like the fear of using this new technology to heal patients? Is there a social bias, the fear of like what might happen if there is a commercialization of embryos?
I think there is legitimate fear that we would use the power of healing as an excuse for the commodification of a class of humans that are produced purely for that purpose, purely for the purpose of healing somebody else. And I think society, to the extent that it would be biased against it, would be rightly biased.
Yeah.
That they would see this as sort of a smokescreen around the real issue, which is, do we have a right as a society to produce copies of ourselves that are fully autonomous, fully dignified, individual, separate humans as an instrument for my advantage, for my healing, for my treatment, for maybe even preventing the effects of aging or repairing an injury that I sustained in a car accident, or any of the other purposes I might want a source of body parts for.
And yeah, that is a crazy topic to talk about. I mean, the use of creating a human life just for your own benefit is a very scary thing. It could definitely become out of line.
Yeah.
Very quickly.
I agree, and I think it's unfortunately, some people even approach children this way. That I'm producing a child in order to have somebody take care of me in my old age. That's perhaps less extreme. You're not using their body parts for your own advantage, but I think exploitation of other individuals has been going on for a long time in human history.
Mm-hmm. So there would be a difference between, let's say, a donation of blood and the use of cloned embryo, because the blood doesn't have autonomy. The blood is created and you're basically giving it away. Unlike cloned embryo, where once the somatic cell is introduced into the egg and produces an embryo that is a newly and individual autonomous person or human life.
Correct. And depending on what the intended use of that cloned individual is, either you're going to actively kill it when it after it has developed to the point when you can isolate medically useful stem cells from that embryo.
Mm-hmm.
So you're producing it in order to gain cells that in a procedure that will destroy that person's life. Or in the more futuristic kind of case of growing up a cloned individual to maturity, you've produced a human being who those individuals who produced it are seeing as property. I made this human so that I could have a heart for transplant or kidneys,
and that's intrinsically wrong. All right, thank you so much. Wow, I just want to give another thanks to Dr. Kondik for joining me on my podcast, and I hope you learned something, because I sure did. During this last segment, I would like to express my own opinions on therapeutic cloning. After reading Dr. Kondik's article and having a conversation with her, my opinions about this topic have solidified. The use of creating a human embryo, even if it's therapeutically cloned, then going on the process of destroying it to benefit another is wrong. I do not believe it will ever be right to commercialize a human being at any stage of human development or life. Following this, the process of obtaining a human egg to attempt the therapeutic cloning surgery can be highly fatal. So it would be unappealing for most women to sign up for this procedure. But if this process of therapeutic cloning was normalized, the need for human eggs would be an increase in demand. This demand would end up being filled by people of a lower economic class, who do not have the economic freedom to deny this risky procedure, because they might believe that the profit of the procedure is worth more than the risk of losing their life. Since the time of this ethical debate, there have been other procedures to help patients who need cell or tissue transplants. So the reasoning to use therapeutic cloning to help people who need tissue transplant can be ruled out because other procedures that are more ethically accepted and less harmful can be used. Thank you to all the listeners of this week's podcast, and I hope you enjoyed our conversation and special interview.